meiosis. So these are formed by a special type of diploid cells that are present in the reproductive organs. Now here, if you take spermatids, okay, primary spermatids, let us take primary spermatids, okay, spermatogonia. Let us take the mother cells of diploid, mother cells of sperm cells called spermatogonia or diploid in condition, okay, having 46 chromosomes. Oogonia in females having 46 chromosomes, hence they are diploid cells and they produces the haploid cells. This kind of division is called as cell division or meiosis cell division. Meiosis ensures the production of haploid phases in the life cycle of sexually reproducing organisms. Now here we comes across meiosis during gametogenesis. If you carefully observe this gametogenesis. Now in plants and animals, this leads to formation of haploid gametes. Now if you carefully observe the key features of meiosis, meiosis involves two sequential cycles, okay, and during this process nuclear and cell divisions called meiosis I generally occurs. In meiosis I it occurs, and in meiosis II, if you carefully observe the meiosis II, during meiosis II only a single cycle of DNA replication generally occurs, okay. Meiosis I is initiated after the parental chromosomes have replicated to produce identical sister chromatids at each S phase. Meiosis involves pairing of homologous chromosomes. Pairing of homologous or equal length sized structured chromosomes are called homologous. And a recombination between non-sister chromatids and homologous chromosomes. And you know that 4 haploid cells, if you carefully observe, 4 haploid cells are formed at the end of meiosis II. At the end of meiosis II. Meiotic events can be grouped under the following phases. Okay? Meiosis I, if you carefully observe. Prophase I of the first meiotic division is typically longer. Okay? Prophase I, it is typically longer, more complex when compared to the remaining phases of prophase mitosis. It has been further subdivided into following phases. Okay, first one is I am talking about the subphases of prophase I of meiosis I. Leptotene, Zygotene, Pachytene, and Diplotene, and Diakinesis. During Leptotene stage, chromosomes become okay, gradually visible under the light microscope. Compaction of chromosomes continues throughout the Leptotene. Okay. Now this followed by the second phase called Zygotene. During this stage, chromosomes start pairing together, and this process association is called synapsis. What is that? Such paired chromosomes are called generally homologous chromosomes. Okay. Now electronic micrograph of this stage indicate chromosomes. Okay. Chromosome synapse is accompanied by formation of the complex structure called synaptimal complex. The complex formed by period of synapses, especially from the homologous chromosomes. So hence this stage is called bivalent or tetrad. Okay. However, these are more clearly visible at next stage. The first two stages of Prophase I are relatively short-lived compared to the next stage called Pachytene. During this Pachytene stage, what will happen? 4 chromatids of each bivalent chromosomes becomes distinct and clearly appears in tetrads. So how this tetrad looks like? The bivalent chromosome generally looks like this. This is called as a tetrad. How many are there here? Chromatids, total 4 are there. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over, this is an area called crossing over, between the non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material. Crossing over is the exchange of genetic material between 2 homologous chromosomes. Crossing over is also an enzyme mediated process. Enzyme involved is called recombinase. Crossing over leads to a recombination. Crossing over leads to recombination of genetic material on the 2 chromosomes. Recombination between homologous chromosome is completed by the end of Pachytene. Leaving the chromosomes linked at the site of crossing over. The beginning of the Diplotene is recognized by the dissolution of the synaptomal complex and the tendency of the recombined homologous chromosomes of the bivalent chromosomes to separate from each other except at the crossover. This crossover helps to exchange the genetic material. The X-shaped structure called chiasmata, what is that? Chiasmata, in oocytes of some vertebrates, if you take the Diplotene, okay, this Diplotene can last for months or years, okay. The final stage of the meiotic Prophase I is Diakinesis. This Diakinesis is marked by terminalization of chiasmata. Finally, during this phase, if you carefully observe the chromosomes are fully condensed. If you take the meiotic spindle is assembled to prepare homologous chromosomes for separation. By the end of the Diakinesis, the nucleolus disappeared and the nuclear membrane or nuclear envelope also break down. Diakinesis represents transition to metaphase. Metaphase I, bivalent, carefully observe the bivalent chromosomes align on the equatorial plate, align at the equatorial plate, okay. The microtubules from the opposite poles of the cell, these microtubules from the opposite poles attach to the kinetochore, as you know very well about kinetochore of homologous chromosome, and pulling them towards the opposite ends. You can see in the diagram in the anaphase one. Okay. Finally, in the cytoplasm, there was a small furrow divides the cell into 2 equal halves with equal amount of genetic material. Hence, 2 daughter cells are formed in the meiosis I. During anaphase I, homologous chromosomes will be separated, while sister chromatids remain at the centromere. Now, telophase I, as I said in the diagram, you can see, although in many cases chromosomes do not undergo for dispersion, but here the stage between the 2 meiotic divisions is called interkinase or interkinesis. What is that? And it's generally short-lived. There is no replication of DNA during interkinesis, but interkinesis is followed by the connecting next phase, that is Prophase II in the meiosis II. Okay. So let us learn about meiosis II in the next coming